Welcome to another tutorial of a video series on genome-wide analysis. In this video, I will take an example of a family of proteins and will show you that how we can find its domain, and using this domain, how we can retrieve and bulk downloads its all member of proteins sequences from different databases, especially rice and Arabidopsis. So let's begin. So for this, first of all, you can create one folder on your desktop. So let me create one folder here. You can see I have already created one folder on the desktop with the name bromo domain video. So because I will be taking example of bromo domain genome-wide analysis. So what we have to do first, we should have one file of Word, and we should have an empty file of Excel, because we will be using them later on for our own analysis, and we will be saving also the text file here. So let me show you that how we can begin. If we want to perform genome-wide analysis of bromo domain family, so first we should have a domain sequence of bromo domain. So it means we should have a bromo domain sequence we should have. Now, so there are the two ways to get the bromo domain sequence. The first way is that we can directly get its sequence from the database. The second, if you found that there is no database reporting your genome-wide analysis protein, so then you can go to NCBI, download any gene that contain that domain, and then using Pfam you can extract that domain. So in this video I will show you that how we can directly download any domains from their database, and from this database we will be using other databases like Arabidopsis and rice to extract other member of this family of protein. So let me open that database. So first of all, you can simply go to Google, and as you know that this bromo domain family genome-wide analysis is already published, so I will be using this to reproduce its results. So the most famous database for domain analysis is used as scope database. So you can simply open the scope database to download any domain sequence. So you can see here is the scope database. Scope stands for structural classification of proteins. So you can simply click on it. So you can see here you can search any domain with free text, or you can also write scope ID, you can write PDB ID or UniProt ID, you can also use any of them. So because we can simply write bromo domain, so then we can simply click on go. So now it will take few seconds to search that what is the sequence of this bromo domain. So here you can see we have actually this bromo domain. So, and if we go to its original literature, you can see even they have reported that which scope database ID they have used as their original search. So we can directly also search this ID to go to this to reproduce the results, because you can see this is also showing this result. So we can simply click on this. You can see now it has fetched all those genes having this domain. So because initially this bromo domain was identified in Homo sapiens, so we can take this. So we can simply click on this. Here you can see this is overall that protein identified and named as bromo domain containing protein. And you can see this highlighted portion is actually that bromo domain. So we can simply click only this part, including this P, and we can take it to that our Word file. So from the database you can see we have identified its sequence, and we can just simply clear its formatting and remove spaces. So now we have actually the basic structure of that bromo domain. So we will be using this sequence as our original search, and we can also confirm the sequence using any other database like Pfam. So what we can do, we can also search its sequence on Pfam bromo domain. Sometimes some databases, depending upon the species, whether you are working on animals or plants, their length may vary up to few amino acids. So we can simply just blast this on the Pfam. We can go here, we can go back, and here search the Pfam. You can see here is Pfam database, and Pfam is now hosted by Interpro. It's okay. So here you can see when you will open this, so it will show you the sequence search. First you can click here, and here you can see it is showing the paste your protein sequence to find the matching Pfam entries. So we can simply search this one, and we can click on this go. It is showing that this one we have entered, so we can click here on again search. So it will take few minutes to search this. So as I have already searched this one, so I can open this. So it means after finishing, your results would be like this. You can see this was actually that domain we have entered. So now this domain is registered with this. In IPR database, this domain is registered with this ID. In PS database and in Pfam database, it is registered with this ID. So we can also simply paste that ID here, because we will be using this ID later on. And we can also know the sequence. You can see this is for this, and it is the sequence from 1 to 33. So we can go here, and from here we can only keep 1 to 33. So because overall this is in the bromo domain database, you can see the overall length is 108, but in this should be around 1 to 33. It should be around half, maybe like this. We can go here and search. This is 31, and we should have actually 33 in Pfam database. But because we will be, you can also use this short form when using this Pfam, but now because if we want to reproduce the results, and that they have actually identified this bromo domain initially from the human beings, so then you can use this as the search. But if you are working on the plants, and in the plants this domain is actually a bit shorter, so you can use this as the shorter version. So I will take simply this, because initially it was identified in the animal database, and in the scope database you can see this database, its length is a bit longer. So now we can go to that RICE database. Here you can see they have mentioned that analysis based on the BRD family IDs using this ID was used for identification of BRD family members, from where using one Arabidopsis database. So we can simply go here and open Arabidopsis database. We can simply go here, use Arabidopsis database, and here is Arabidopsis database. So first of all, we can go to tools, and we can go to this blast search, and here you can see we have different options for blast program. Here is blast nucleotide, here is blast proteins. So because we want protein, so we can simply select here this blast protein, and we can also select the database, because now nowadays they are releasing new databases. So although you can use this tier 10 proteins, you can also use this latest one. So I will go with the latest one, and here is our protein query sequence. So we can simply click here and go to this domains, and we will be finding those all genes which are in Arabidopsis and have this domain. So we can simply click on this run blast. It will take few seconds. Yeah, you can see here we have blast results, and you can see almost 23 sequences out of 50 they have just mentioned. And here you can see here we have different IDs which have this domain. So what we can do, because in the Arabidopsis genome database you cannot directly export this data. First you should have the sequence of this all list. So what we can do, we can simply copy these all sequences, and we can extract only these IDs, and later on these IDs we will be extracting their protein sequence. Here one thing important to note, that you can see in the first one, I can little zoom in. If you see this AT5G 550040.2, this and this gene is actually coming twice. So what it's mean, actually the gene is one, but its transcript is two times. I mean there are two types of transcript of this gene. So in the final version we will be only selecting one. So here you can see other one like this gene has even 1 2 3 4 5 6 transcripts. So what you will do, you will only take it as a once, and other you can write that this gene has one transcript, two transcript, like four transcript, like this they have done here. Let me show you how they have done. You can see here, like the first gene, they said it is located on the chromosome number 1 and the number of transcript is only 1. But here you can see for this, you can see the number of transcript are 4. And similarly you can see for this gene the number of transcript are 6. So you can only mention that gene for once, and but you can show that this gene has this number of transcripts. So hopefully now this question is clear. Now we will be only extracting this gene sequence. So what we can do, we can simply go to this copy, and as we have already created one Excel file, so we will be using this. We can simply paste it here. As you know, we only need this first column. So what we can do, we can go to this data tab and we can convert text to columns, and we can delimit with the space, because here was space. So you can see now we have actually that list. As you know, now we have this column. So we will be directly, if you can see, if we will copy this to our Excel file, so it will show you a string list in the form of this table. So to avoid this, what we can use another website, that is, we can open to Google and convert tableconvert.com. tableconvert.com. Yeah, you can see here we have this website. This website we can freely use, and we can simply only paste here. When you see when we have pasted that file here, it is showing in the form of table, which will take too long space. So we will first simply paste in this so that we can simply copy all this. And the benefit of this website is that we can remove duplicates here, we can delete empty blanks, deduplicate, transpose. This website is actually pretty useful. So now we can, as you know, we have simply copied this already, so we can now prepare our list of Arabidopsis. List of genes from Arabidopsis genome. Here is that list. So now what we need to do, we need to actually bulk download their sequence of amino acids or proteins. So what we can do, we can simply copy this, and we can go back to this BLAST database. We can go to home, you can see, and simply we can use this another tab. Let's zoom in back. We just need to reset, and here you can see, if you go to this download tab, here you can see there is bulk data retrieval. So we can go on here, and here we have actually the sequences. So we can simply click on this sequences, and here we can just only paste those IDs, as I have already copied that gene from my list. And here it is asking what you actually need, what you need as transcript, or you need as coding sequence, you need their genomic sequence. If you also want to show the gene structure, so then you can also go to this genomic locus sequences. But if you only want their protein sequence though, so then you can only select this protein sequence in the form of FASTA. So that's it. So you can simply click on here as get. So here you can see now we have a list of all those genes having their protein sequences. What we can do, we can simply copy all this, go to our main folder, and we can here create one text file. So we can create, this is bromo domain genes in Arabidopsis. We can simply copy here. Let me finish the previous files. Yeah, we can simply paste here. As you know that now this actually has more names. You can see this is actually the FASTA format. As you know that the FASTA format begins with this greater than sign, and we should have only this gene name for the simplicity, and all these other things we should remove, and this termination codon we also need to remove. So there are two ways. One way is that you can do manually, but as you know, it will take too long time. So what we can do, we can simply just copy all this, and we can go to that software which I have already told you about for my first, second, and third video when I was telling about databases. So this is actually the TB tool. So you can go to this TB tool. Here is this TB tool. We can go to this sequence toolkit. We can go to this FASTA tools, and here you can see ID simplify. So we can click on this. So it is showing now here we can use this for ID simplify. Here are the two tabs because we want sequence as input. So we just simply pasted that sequence what we got from there. And our purpose is just to remove other things except this gene name. So we want input here, so we can simply click here. You can see the congratulations. You can see congratulations simply finished and the output info was placed. So you can see now this gene has all these things are automatically removed. So it is pretty helpful. So we can go select all, or other way you can just use Control A to copy, and we can create one more file here, new file. We can write here bromo domain from Arabidopsis thaliana simplified. We can name it as simplified. We can open this and we can paste here. You can see now their genes have only gene IDs, so we can simply use this one. As in this list, you can see now we have simplified protein sequence. So we will be using this sequence list later on for our other videos and for later on analysis. So if you carefully notice that here the random gene IDs, like here they are just the simply gene IDs. It should be like Arabidopsis thaliana BRD1, it should be 2, it should be 3, because we have to name them in the sequence, but this part we will do in our later videos. So here our bromo domain from the Arabidopsis thaliana genes is ready. So now let's move to the RICE database. So as you know that we have already prepared this list having bromo domain genes from Arabidopsis. So now we will search this amino acid sequence from the RICE database. So here in this research article, you can see they have used this RICE database. So we will be using this RICE database again to get sequence of we can use this RICE database. So for this we can simply go to this search function and use this blast search. So one way is that we can simply use this blast protein function and we can enter here that sequence of bromo domain and we will search using this. We can change the overall number of alignments to maximum so that it can get, and we can simply click on this submit button. But actually this RICE genome annotation project has another beautiful function. What is that? That we can simply search any list having the domain search, and you can see we can simply write only the Pfam profile. Here you can see the second way is that Pfam profile. As you know that we have actually this Pfam profile ID. So if we will write only this ID, so we don't need to paste the sequence one by one. So we can simply write that we want to filter all those genes having this Pfam profile domain. So we will simply click on this search. So here you can see the list is populated having all these Pfam profile. So we can simply click on here, because in this list you can see we have model gene ID, then we have putative function, Pfam ID, then Pfam name, that is bromo domain, and their e-values. But actually we want to retrieve their sequence as we have retrieved for Arabidopsis thaliana. So what we actually want, that we want this list. So to generate this list, we will go to this website again and we will search this. Here you can see this table. We actually need this second column only for this list. Here you can see though now that list is prepared. We can copy it again. We can go here again and paste that list here. So you can see now there would be no space in between. So now we have a list of genes from rice. So now we want to retrieve their sequence. We can simply copy this one. We can go to this rice database again, and we can go to this download option, and to download option we can click on this genome wide annotation batch download, and here you can see it shows please select the data type you wish to retrieve. We wish to retrieve protein sequence now, but if you want, again, like as I have already said for Arabidopsis, that if you want to report the gene structure, then of course you can identify and download the genomic sequence as well, but for now we are only retrieving the protein sequence, so we will be only retrieving the protein sequence for this gene. So we can simply click on this submit in the form of plain text. So here you can see now our list having the protein sequences of all these genes from the rice is also ready. We can simply copy it. We can go here again. We can make it like bromo domain from Oryza sativa, and here just paste this one, and we can only simply save. As here you can see now this name is extra in this. You can see this is actually the gene ID, and here is the FASTA format because it begins with greater than sign. This word is extra, although we can use it using our control H function, but I will go again with that as we have done for Arabidopsis. We can go to this TB tool and we can use simplify ID function. We can go here, we can go to FASTA tools, and we can go to this ID simplify. You can see here we can paste input sequence, and here we can get the output sequence. Here you can see congratulations, simplify finished, and output info was replaced in this output box. So now we can copy this one and go back to our folder, and here we can write bromo domain from rice Oryza sativa, and we can write simplified. And here, as we have done previously, you can see there is actually this termination codon. We can simply use Control H and replace this star. You can simply write here star and replace with nothing. You can press on this replace all, so you can see this termination codon is automatically removed. So we can simply save this. So now our two lists are ready that we will be using in our later on videos. In the later on videos we will be just renaming them and use it for our own later on purposes, like we will use them for phylogenetic analysis. But before that, this should be like in the sequence 1 2 3, but that thing we will do in later on our videos. So far we have generated the list. As they have generated the list, if you can see in their results, so here they have list in the table 1. This is the list from the Arabidopsis thaliana, and similarly here they have a list of from rice or Oryza sativa. So now our lists are ready. So you can also use other databases by using this same function. You can blast your amino acid sequence and then go to bulk download. Sometime If your species has no specific database, then of course you would be using NCBI, but for most of the plant species, they have their own databases, so we will directly use them. So hopefully this video will be helpful for retrieving amino acid sequences from different databases and how we can simplify that. So far, if you have any question related to the data retrieval, please let me know in the comments section. Thank you for watching. Bye bye.